Good morning everyone. So for today's video, I want to discuss to you about the microbial structures. But before that, don't forget to subscribe my channel. So we have heard the cell morphology. Bacteria have characteristic shape. The common microscopic morphologies are spherical or ovoid cocci, the red shape for bacilli, and the comma shape for vibrio, and spiral for spiralium and spirochetes. Some cocci characteristically grouped in pairs of chains, and some from group-like cluster of spherical cells. Some round cocci form cubic pockets. Bacterial cells of other species grow separately, and the microscopic appearance is therefore valuable in classification and diagnosis. So we have heard the structures of bacterial cells. The protoplasm of the whole body of living materials is bounded peripherally by a very thin, elastic, and semi-permeable cytoplasmic membrane. Outside and closely covering this lies the rigid, supporting cell wall, which is porous and relatively permeable. So we have heard the cell wall. Cell wall encases the protoplasm and lies immediately external to the cytoplasmic membrane. It is 10 to 25 nanometer thick, strong, and relatively rigid, though with some elasticity and open porous by being freely permeable to solute molecules smaller than 10 in mass and 1 nanometer in diameter. It shows granular structures and lacks microfibrils. We have heard the gram-positive and gram-negative. Bacteria have different type of bacterial cell. Gram-negative and gram-positive is different of this wall. Gram-positive type wall is very thick and generally has simple uniform appearance under the electron microscope. Some 40 to 80% of wall is made of tough, complex polymer and peptidoglycan. In this type of wall, the succulus consists of multi-layered peptidoglycan which during growth develop by the inside and outside mechanism. The gram-negative or the negative gram, the cell wall of negative gram or gram-negative bacteria are more chemically complex, thinner, and less compact. Peptidoglycan makes only 5 to 20% of cell wall and is not the outermost layer. The peptidoglycan of gram-negative bacteria is located between the plasma membrane and outer LPS membrane. The LPS membrane is similar to a plasma membrane, but less than permeable and is composed of lipopolysaccharides or LPS. We over the plasma membrane. Plasma membrane is a bacteria are composed of phospholipids consist of polar group attached to a 3-carbon glycerol backbone. There are also 2 fatty acid chains dangling from the other carbons of glycerol, the phosphate end and the molecules of hydrophilic and is attached to water. The fatty acids are hydrophobic. Membrane are also contain proteins. Typically 20 to 30% membrane, most of them are placed in the membrane so that the hydrophobic amino acid associate with the lipids in the membrane and the hydrophilic amino acid are outside the membrane interacting with either the cytoplasm. We have heard the function of plasma membrane. The plasma membrane is selectively permeable in that helps control what moves into and out of the cell. Another one is extracellular structures, the fimbria or the pili. Pili are elongated or hair-like proteinaceous structures which protect from the cell surface. They are specifically on those gram-negative cells. Fibria are typically 2 to 3 nanometer in diameter and from 0.1 micrometer to several micrometer in length. They may occur all over the cell or may localized. Flagella. The flagella are made up of glass and linear protein class called flagellants. The flagella, which arise at various points of the cell surface. Flagellated bacteria are described as monotribous, and the leukotrichous, and the pitritrichous. It's depending on how the flagella are arranged. The capsule is lime layer. The capsule of most bacteria consists of polysaccharides. The bacteria of a single species can be classified in different capsules, spirals, or serotype based on the fine chemical structures of these polysaccharides. Another one is intracellular structures. We have the plasmids. Plasmid was the first introduced by the American molecular biologist named Joshua Lederberg in 1952. A plasmid in short using cellular and double-strand segment of DNA that is found in a cytoplasm separate from the main bacteria chromosomes. Their size varies from 1 kilobase pair to over 100 kilobase pair. Plasmids are capable of autonomous replication. Plasmid can transfer genes from one cell to another cells. We have heard the ribosomes. The cytoplasm contains a large number of solute, low and high molecule weight substance, RNA, and approximately 20,000 ribosomes cells. Bacteria have 70 ribosomes, comparing 30s and 50s subunits. We have heard the function of ribosomes. The functions of ribosomes as the organelles for protein synthesis. The mesosomes are covulated or multilayered or multilaminated membranous bodies visible in the electron microscope. We have heard the function. Functioning in the compartment of DNA at cell division and at sporulation, having a function analogous to the eukaryotic cell, providing a cell membrane support for respiratory enzyme. The last one is that nucleoid. The nucleoid is a region of a cytoplasm where where the chromosomal DNA is located. It is not a membrane-bound nucleus but simply an area of the cytoplasm where the strands of DNA are found. Most bacteria have a single cellular circular chromosomes that is responsible for replication, although a few species do have two or more smaller and auxiliary DNA strands called plasmids are also found in the cytoplasm. So those are the part of microbial structures that I discussed to you. So that's all. Thank you.